Hello students, welcome to Vivid Botany. We have a question on central dogma of molecular biology. Let us see dogma. Dogma means principle, that is a doctrine, a rule laid down. So, the central dogma of molecular biology. It is proposed by Sir Francis Crick of biology, proposed by Francis Crick in the year 1957. He also the first one to term it central dogma. What does it mean? It is something like core technique, something like core principle of biology. What does it mean? We know that master chemical is DNA. When DNA double helical structure was discovered, when DNA was found to be the genetic material of all organisms except some viruses, the DNA and many other concepts about the DNA were revealed, among which one of these is central dogma of molecular biology. So, basically speaking, DNA has got two functions. That is, DNA can give rise to two DNAs, two DNA molecules. It can give rise to two DNA molecules. And same DNA can give rise to mRNA, that is messenger RNA. The former one is called as replication or duplication or synthesis or making copy of the DNA. One DNA gives rise to two DNAs. What is the necessity of one DNA giving rise to two DNAs? See, in the mitosis, one mother cell gives rise to two daughter cells. The two daughter cells receive the same copy of DNA whichever is there in the mother cells. So before the two daughter cells are formed, before the mitosis, the DNA present in mother cell undergoes replication and two copies are formed, and the two copies are transmitted, inherited into the two daughter cells. If the two daughter cells give rise to four daughter cells by another mitotic division, the same replication takes place in the two daughter cells. They give rise to four DNAs. Four DNAs are transmitted into four daughter cells. Like that, from zygote, any number of daughter cells are formed during embryogenesis and during the body formation. Zillions of cells are formed. In all these cells, the same copy of DNA is inherited into all these cells. That is, often we say that zygote is what do you call that? The first cell of the whole living body. The whole living body is formed from the zygote. So whatever DNA is there in the entire body, the same DNA present in the zygote itself. For that, the main thing is DNA replication or DNA synthesis is involved. That plays greater role. Let's have a look at the formation of mRNA. This formation of mRNA is called as transcription. It is done by an enzyme called as transcriptase. That's what today call it as, nowadays we call it as DNA dependent RNA polymerase. DNA replication is catalyzed by DNA dependent DNA polymerase and transcription is catalyzed by DNA dependent RNA polymerase. Now, from this mRNA, proteins are formed. This process is called as translation. So here again many enzymes are involved like peptidyl transferase or ribozyme or amino acid synthetase, like many are involved here. So formation of proteins from mRNA is called as translation and formation of mRNA from DNA is called as transcription. This is called transcription. Transcription translation, both are the unidirectional flow of genetic information from DNA to mRNA, mRNA to protein. It is visualized, it is understood as unidirectional flow of genetic information from DNA to mRNA and from mRNA to proteins. So this information which is flowing from DNA to mRNA, mRNA to proteins, it is usually irreversible. It is not reversible except in some cases. So we should understand that proteins are made up of amino acids. DNA is made up of nucleotides. Proteins are made up of 20 amino acids. DNA is made up of 4 types of nucleotides. That is, the sequence of amino acids in protein is decided by the sequence of 4 nucleotides in DNA. That is, the types of proteins, the amount of proteins, the quantity of proteins, all that are determined by the DNA. In fact, in our body, all the proteins are formed from the same DNA. I mean, the DNA has got that much information that it can form all types of proteins that will make up one's body. So often we call it DNA as the master chemical. So this is a core technique where DNA plays the major role and the prime role, the most important role in forming the biochemical compositions, namely the proteins. So these proteins may act like enzymes, hormones, antigens, antibodies, drugs, toxins, whatnot. Many things are formed from these proteins. And these proteins are formed from mRNA. mRNA is formed from DNA. Why mRNA is formed here? Because DNA cannot come out of the nucleus. The translation process takes place in cytoplasm. Because proteins are made up of amino acids. Amino acids are present in cytoplasm. Those amino acids are constructed to form the proteins with the help of the coding information present on mRNA. So mRNA carries the genetic information required for synthesizing protein in cytoplasm. mRNA is made inside the nucleus from DNA, from where it is transported to cytoplasm to form the proteins. So the whole molecular biology or the chapter that we study, molecular base of inheritance, will revolve about only these two lines, that is central dogma of molecular biology. Okay, so DNA has got two functions. DNA gives rise to DNA. Other function is called as DNA gives rise to mRNA. mRNA gives rise to proteins. And formation of DNA from other DNA is called as autocatalysis also. Autocatalysis also it is called as. So it has got DNA replication, DNA duplication, DNA synthesis, DNA autocatalysis. And formation of mRNA from DNA is called as heterocatalysis. This is heterocatalysis. So both are well explained, well understood in Central Dogma of Molecular Biology. And one more interesting thing is that sometimes a DNA may form from mRNA. This mRNA may give rise to DNA. This DNA often we call it as complementary DNA. It is a single stranded ssDNA which is complementary DNA we call it. And formation of complementary DNA from the RNA is called not exactly mRNA but from RNA. I write here, from RNA the cDNA is formed. It is called as reverse transcription. It is called reverse transcription. And for this enzyme is reverse transcriptase. The enzyme that the catalyzed here is that reverse transcriptase, and it is discovered by Temin. So we call it as Teminism. Teminism, do remember, it's also asked many number of times. This takes place in retroviruses, right? So in viruses this phenomena is very common, formation of complementary DNA from RNA. So this is the Central Dogma of Molecular Biology proposed by Sir Francis Crick in the 1957 and published in the year 1958. Now let us see the options here. A is here formation of DNA from DNA. So this must be the replication. Replication is given in option 1. Then B is transcription. B is given transcription in option 1 as well as option B also. So 1 and 3, 1 and 3. Then C is translation. See, C is given here transduction. What is transduction? It is something related to the viral multiplication. That is, the virus will carry the genetic information, the DNA string from one bacteria to other bacteria and causes, it acts as a vector, it acts as a vehicle of carrying this DNA strand from one bacteria to other bacteria. It's called as transduction. It is proposed by Lederberg Ginder. But here formation of the protein from mRNA is called as translation. So this is the right answer. First one is replication, second one transcription, third one translation, and the D is protein. So this is third option, the right one, Central Dogma of Molecular Biology. So I think 3 to 4 questions are extracted from molecular biology. So it appears to be very much important since it is the core chapter of the molecular biology, which will lay foundation for all the future courses that you adopt in your future career. So take care of the chapter very much and don't forget to subscribe my channel Vivid Botany. We will be tackling all the NEET 2020, 2019, 2021 questions and analyzing all the concepts whatever are covered in that question very in detail. In detail we can cover that. So thank you very much.